Yes, boys. In histology, today we will talk about the excretory system. The function of excretory system is removal of metabolic waste products from blood to exterior. As I have used metabolic waste products, the metabolic waste products they are converted into urine by the kidney. All right. So the respiratory system is consisting of pair of kidneys, pair of ureter, urinary bladder, and urethra. As I told you, the function of excretory system is to excrete the metabolic waste products. The metabolic waste products which are present in the blood are converted into urine by the kidneys. This urine is excreted out through the ureter, comes into the urinary bladder, and from the urinary bladder it comes out through the urethra. So all these are the parts of excretory system. Besides the excretion of the urine, the excretory system also having other functions. The other functions are it also regulates the fluid and electrolyte balance of the body. Thirdly, it also secretes the hormones. Hormones secreted are renin and erythropoietin. So these are the three functions of the kidney: formation of the urine and excreted out. Second, it also helps in the maintenance of the fluid and electrolyte balance of the body. And thirdly, it also secretes the hormone. The hormones secreted by excretory system are the renin and the erythropoietin. The diagram you that is the gross picture of the kidneys. As during the time of peritoneum, I had told you the primary retroperitoneal organs and secondary retroperitoneal organs. Kidneys are also the retroperitoneal organs. They are situated in the posterior abdominal wall in the region of lumbar region. Each kidney, they are bean shaped. Look here. The kidneys are bean shaped structures, and these bean shaped structures on this is the lateral wall, this is the medial wall. The medial wall of the kidney is slightly depressed, and that part is called hilum of the kidney. That is called hilum of the kidney. Through the hilum of kidney, the structures entering are the renal artery and excreted structures from hilum. One is the renal vein, another is ureter. I had told you two things: structure entering in the kidney through the hilum and structure excreted out from the kidney through the hilum. The structures entering are the renal artery. Structure excreted out or coming out from the hilum are renal vein and the ureter. You are seeing here the dilated part. This is the dilated part of the ureter, and this dilated part of the ureter is called the pelvis of the ureter. Both these ureters, they are opening into the urinary bladder, and from the urinary bladder they are passing the urine through the urethra. The urethra differs in both male and female on the structural aspect that I will tell you later on. So, in the beginning I had told you the three functions of the excretory system: formation of the urine, that metabolic waste products are converted into urine by the kidneys, and this urine through the ureter comes into the urinary bladder, where they temporarily store, and on their will they excreted out through the urethra. Each kidney that they are I am telling you pictures. If we take the sagittal section of the kidney, you know the sagittal section of the kidney, that suppose this is the kidney. If we take the sagittal section, I will show you in the dissection hall also, taking the cross section of the kidney. Okay. When we see the sagittal section of the kidney, what we find that kidney is divided into two parts, outer cortex part, inner medullary part. Look this diagram. This is the cortex part and this is the medullary part. When we see the with naked eye, what we find that cortex part that is not showing any structure with naked eye. I am using the word with naked eye. While the medullary part can easily visualized by the naked eye, that it is showing the pyramids. These pyramids are about 10 to 15 pyramids in the medulla of each kidney. The pyramid is having the dilated part, that is base of the pyramid, that is facing towards the cortex, and this is the apex part of the pyramid. The apex part of the pyramid, you are seeing these structures, these are called the papillae, and they are of different type. Then these papillae, some are the smaller and some are dilated, or you can name this as calyces. So the calyces which are smaller in size, they are continued with the apex of the pyramids. So these are the minor calyces, and as they go down, these are the major calyces, and these major calyces, they are opening here in this sinus. So what is the sinus? Renal sinus, that is a cavity. You are seeing this cavity, and this cavity is continued into the pelvis of the ureter. So the thing is that urine is formed here. In the each pyramid there is present the tubules. The tubules are different size, collecting tubules, distal tubules, all that. So through these tubules the urine comes into the minor calyces, and then major calyces. From the major calyces it comes into the renal pelvis and then excreted out. That is the gross picture which we can see by the naked eye also. Histologically, when we see that, what we find that cortex part of the kidney is consisting of renal corpuscles, and the medullary part is consisting of the tubules, different type of the tubules and all that. Let's see what is the renal corpuscles. Look this diagram. This is the red color is the top vessels, capillaries. What happens? That renal artery enters into the descent of the capsule. So the point through which the renal artery enters, that is called the space, blood space, through which, or vascular pole of the capsule, and on the another pole you are seeing the another structures. These are the proximal convoluted tubule, and this is called urinary space. Alright, the second thing, that capsule is consisting of double layer, and this double layer part is called Bowman's capsule. That is called Bowman's capsule, and this Bowman's capsule is surrounding this top capillaries, that is called glomerulus. So both these structures, glomerulus surrounding by the Bowman's capsule, is called the renal corpuscles. This structure. After seeing this, now When we talk about the kidney is consisting of million of nephrons, and these nephrons are the structural and functional unit of the kidney. Each kidney consisting of million of nephrons, and these nephrons are the structural and functional unit of kidney. There is no regeneration of the nephron. First thing. What is the nephron? Nephron is consisting of these things as shown in the diagram. First is the renal corpuscles. The renal corpuscles, as I told you, consisting of Bowman's capsule and tuft of capillaries, and having the two species that I will tell you just now. First of all, see what are the different parts of the nephron. First is the renal corpuscle. Then it consisting of tubules. The tubules are of different type. First is the proximal convoluted tubule, PCT. PCT starts from the beginning of the urinary pole of the renal corpuscles. So this is the proximal convoluted tubule. Then you are seeing this U-shaped structure. This U-shaped structure is loop of Henle. Then this loop of Henle is continued into yellow color structure. This is the distal convoluted tubule, yellow structure. This distal convoluted tubule is continued into the collecting tubules. And these collecting tubules are opening into the larger structures that is called collecting duct. All these are the structure of the nephron. Understand? And these nephrons are million in number, and there is no regeneration of the nephron. Now we will talk one by one of these structures histologically. Look this diagram. When we focus the slide under the microscope of kidney, you will find these structures. Cortex part, and this is the medullary part. The cortex part is showing the large number of the renal corpuscles. These are the glomerulus surrounding by the Bowman's capsule. One thing. Another thing, we will find the large number of the proximal convoluted tubule, PCT. Second, there is also present the distal convoluted tubules. So all these are structures present in the renal cortex. Cortex part of the kidney is showing all these structures, and it is asked during exam, what are the structures present in the cortex part? In cortex part, you have to speak that renal corpuscles, proximal convoluted tubule, distal convoluted tubules, these things. In the medullary part, presents the part of the collecting tubules, collecting ducts, and some part of the distal convoluted tubule also. So these are the structures which are seen in the microscopic picture of the kidney. Now discuss what is the structure of renal corpuscles and what is the structure of proximal convoluted tubule, loop of Henle, distal convoluted tubule. Let's see the renal corpuscles. See this diagram. This is the tuft of capillaries, and it is consisting of the two coverings, Bowman's capsule. Outer layer is parietal layer. The parietal layer is consisting of, I have given the example of parietal layer, while the epithelium is squamous type of epithelial cells. So parietal layer is consisting of squamous epithelial cells. AC नहीं चल रहा क्या? No? And another layer is surrounding this tuft of capillaries. The layer which is surrounding the tuft of capillaries, that is called visceral layer. The visceral layer, books are mentioning consisting of podocytes, but we are not able to see the podocyte in light microscope. They are visible only under the electron microscope. So you just speak parietal layer and visceral layer. The space seen between the two, that is this space, This space is called the urinary space where the urine is collected. Okay. As I told you previously that renal corpuscles is having the two ends. One is the vascular pole through which the renal artery enters. Another is the urinary pole from which the proximal convoluted tubule begins. Alright. As I showing you this diagram, look here. This is the part of distal convoluted tubule. This is the part of distal convoluted tubule, and these are the afferent arterioles. These are the afferent arterioles. I will use this word again. See this diagram carefully. At the vascular pole of the renal corpuscles, the afferent arteriole, that is this one, and close to the afferent arteriole, this is the distal convoluted tubule. The thing which I want to tell you here, at the vascular pole of the renal corpuscles, the distal convoluted tubule, the part of the distal convoluted tubule which is close to the afferent arteriole, is showing the different type of epithelial cells. Normally, the distal convoluted tubule is lined by cuboidal epithelial cells. Listen carefully. Distal convoluted tubule normally is lined by cuboidal epithelial cells. You can see here. दिख रहा है आपको? See the diagram. This is lined by cuboidal epithelial cells. But the part of distal convoluted tubule which is close to the afferent arteriole, the cells are columnar. Have you noticed this difference? The part of distal convoluted tubule close to the afferent arteriole, the cells becomes columnar, and these columnar cells of distal convoluted tubule is called macula densa. So this is the macula densa. This macula densa will come again. It is sensitive to the sodium concentration in the blood. Macula densa is sensitive to sodium concentration. So it is clear what is the macula densa? Alright. See the afferent arteriole. You are seeing that the smooth muscle fiber, I am using the word smooth muscle fiber of afferent arteriole, close to the macula densa. Close to the macula densa is comparatively taking the dark stain. This one. And this darkly stained smooth muscle fibers which are close to the macula densa are named as JG cells, juxtaglomerular cells, which are secreting the hormone renin. And third, you look between the afferent arteriole, distal convoluted tubule, you are seeing the another extra cells. These are the Lasik cells. The function of Lasik cells is not clear, but it is believed that they are responsible for the erythropoietin hormone. What is the function of erythropoietin? They stimulate the bone marrow for the erythropoiesis. They stimulate the bone marrow for the erythropoiesis. Alright, now come to the EG part. The proximal convoluted tubule, when we see, look this another diagram of the renal corpuscles. This is the tuft of capillaries. The interrupted line showing it is the visceral layer and the squamous epithelial cells on the periphery, and this is the urinary space. And this is the another end, the urinary pole, and this is the vascular pole. Afferent arteriole enters, they divide into the capillaries, and these capillaries are continued into the efferent arteriole. Alright. Now I am coming to the PCT part. The PCT begins from the urinary pole of the renal corpuscles. And this proximal convoluted tubule, which are present very large enormous number in the cortex part of the kidney, they are lined by cuboidal cells. but keep one thing in your mind. The cuboidal cells of proximal convoluted tubules are showing the microvilli. Look here. You can see also here. They are lined by the cuboidal cells when they are showing the microvilli. Distal convoluted tubules also lined by the cuboidal cells, but no microvilli. This microvilli increase the surface area of proximal convoluted tubule. The function of the proximal convoluted tubule will ask you what is the function of proximal convoluted tubule, what is the function of DCT, and all that. So you must be very clear about this. The function of PCT is absorption of the sodium and water. Absorption of the sodium, water, also the bicarbonates, chlorides, amino acids, and glucose. That is the function of proximal convoluted tubule. Then see the part of distal convoluted tubule at present, because the diagram is showing very clearly. The distal convoluted tubules also lined by the cuboidal epithelial cells, but they are not the microvilli. See this one. They are not lined by microvilli. The function of distal convoluted tubule on the influence of hormone aldosterone. It absorbs the water and sodium that helps in the concentration of urine. About the part of the loop of Henle. See the diagram. The loop of Henle is having the two parts, thicker part and thinner part. The loop of Henle is lined by the cuboidal cells in the thicker part. The thicker part of loop part of loop of Henle. So thick part of both, whether the ascending limb or descending limb, they are lined by the cuboidal epithelium, while the thinner part of both, they are lined by the squamous cell. The function of loop of Henle, it is increase the osmotic gradient of the renal tissue, and this increasing osmotic gradients will help in concentration of the urine. Very simple thing. The loop of Henle is increasing the osmotic gradient, and this osmotic gradient helps for concentration of the urine. About the distal convoluted tubule, as I had told you, that is lined by cuboidal epithelial cells, except the part of DCT which is close to the afferent arteriole, that is showing the columnar cell and called it macula densa. Collecting tubules and collecting ducts. The collecting tubules are again lined by the cuboidal epithelial cell. Collecting ducts may be cuboidal or columnar. The function of collecting tubules and collecting ducts are for the concentration of the urine. That is the picture of kidney, but it is not sufficient. Have you been taught in physiology, respiratory system? Yes? मुंह से बोला करो yes. Don't. All right. Tell me. When the blood pressure is low, when the blood pressure of the person is low, what happens? blood pressure is low, so sodium concentration will be low. And sodium concentration low will stimulate the macula densa. Once the macula densa will stimulate, it will stimulate the JG cells. And JG cells stimulated, they are acting over the angiotensinogen. Angiotensinogen कहाँ से आ रहा है? You have read in the physiology. I forget that you people are not responding any answer. From the liver. So, JG cell acting over angiotensinogen. And what the JG cell? Renin. So what happens? As the sodium concentration is low, they stimulate macula densa. Macula densa stimulates JG cell to secrete the hormone renin. And this renin is acting over angiotensinogen. And that angiotensinogen is converting into angiotensinogen 1. Right? This angiotensinogen 1 is converting into angiotensinogen 2 by the converting enzymes which are coming from epithelium, endothelium of the lung capillaries that convert the angiotensinogen 1 into angiotensinogen 2. Now this angiotensinogen 2, where it is acting? अच्छी तरह बोलो, नहीं मालूम? On the glomerulus of suprarenal glands. On the glomerulus of the suprarenal gland and secreting the hormone aldosterone. This aldosterone is acting over the distal convoluted tubule and absorb the sodium and water. Absorb the sodium and water, and that increase the blood pressure. All right. So remember this in a cycle. I cannot draw here the cycle otherwise. Starting from low blood pressure, stimulate macula densa. Why? Because sodium concentration sensitive macula densa. From this macula densa, this converting low pressure stimulate the JG cells secreting the hormone renin, and renin is acting like this. So draw in a cycle so you can remember. That is the function of the kidney. So you can say that it also helps in controlling the blood pressure. Okay? Any problem? Not yet. And while in the gross, when you will read the anatomy, gross anatomy, see the kidney. The blood supply of the kidney is not easy. And question may ask you, whether it is asked in the university exam or not, but I will ask. I am telling you first. Afferent arteriole enters into the kidney. These afferent arterioles dividing into the first, and these arteries, they further dividing into the smaller branches. And these smaller branches, as they comes at the junction of corticomedullary junction, that is this part, corticomedullary junctions, they are giving the arcuate arteries. Arcuate arteries running over the surface of the pyramids. From the arcuate artery, the another branches arise, and all these they are forming the plexus, and these plexus are present in the Bowman's capsule. I have no diagram at present. I delete that. Otherwise, I will show you the blood supply. Then come to the another slide. Ureter. Very easy to identify. I never noticed that none of the students identify. Every student identifies this slide. But problem arises on the YOC. When we focus the slide, you will see this picture, star-shaped lumen. star-shaped lumen surrounding by the lamina propria part and the muscular layer. Okay. The point is this, that ureter, urinary bladder, and some part of urethra are lined by transitional epithelium. Now I will not tell you what are the structures present in transitional epithelium. Previously I had told you that when we talk of the systemic part, revise the general part. So while drawing the diagram of excretory system, ureter and this, one on the side you draw a small diagram of transitional epithelium. So it will keep retained in your mind. Lying over the basement membrane, the columnar cell, one. Above the columnar cells, there is layer of polygonal cells. The layer of polygonal cells never draw less than 3. Keep this thing in your mind. Never draw less than 3. It may be 5 to 6. If the bladder is empty, or it may be up to the 5 to 6 layer. In case of bladder full, then the layer will depress and it becomes 3 layer. Okay. On the superficial cells, yes, you are speaking correct. But tell me, what is the difference between dome facet shaped cells and cuboidal cells? बोलो, आपसे ही पूछ रहा हूँ. No doubt, you are right. On the superficial surface, there is the dome facet shaped cells. You are drawing the cuboidal cell. Length and width are equal. Nucleus is in the center. All right. The difference between the cuboidal cell and dome facet shaped cell. In dome facet shaped cells, the inferior surface or inferior line of the cuboidal cells, you make it arch shape. Umbrella hota hai. Some books mention it as umbrella cell also. Why? The inferior surface of the umbrella is arch shape. So make the arch shape inferior surface of cuboidal cell. That is dome facet shaped cell. Why it is arch shape? There is reason. When the epithelium will compress in the case of distension of the bladder is full, then the layer will compress. So this inferior arch shaped surface provides the space. Okay. Because of that, it is dome facet shaped cell. So that is the transitional epithelium. And transitional epithelium will show the star shaped lumen. So you will easily identify that this is the transverse section of ureter. This is the lamina propria part showing the blood vessels, lymphatics and nerve fibers also. Then the muscle coat. Look the muscle coat. Inner longitudinal. I am using the word inner longitudinal. Outer circular. What is the muscular coat? What is the muscular coat in GIT? हेलो. हाँ तो पहुंच जाना घर पे यार. अरे मैं नहीं हूँ, घर पे सर्वेंट है. ठीक है. I am asking you, the muscular pattern in the ureter is reverse to the GIT. Here, the inner longitudinal, outer circular. In GIT, the inner circular, outer longitudinal. Okay? So keep this thing in your mind. Underline that. Because these are the things, once you identify the slide, reply well, the next question will be, what about the muscular coat? If you do the mistake, then you will get the normal marks. I am telling you very clearly. That is my criteria to give the marks. Then, all right, the adventitia part, that is consisting of loose connective tissue. हो गया? No. The next question is, the section of ureter is from which part? Upper part, upper two-third part, or lower one-third part? There is a difference. If the section of ureter is upper two-third part, the muscular coat is showing this picture. Only the difference by the muscular coat we can distinguish whether the section is upper two-third part or lower one-third part. If the section is taken from lower one-third part, then there is extra longitudinal muscular coat, outer one. So there will be the three coats: inner longitudinal, middle circular, and outer longitudinal muscle coat. So if you find the muscular coat in three layers, inner, middle, and outer, that means the section of ureter from lower one-third part. Clear? If it is showing only the two layers, that means the section of ureter from upper two parts, upper two-third parts. Then we come to the next, urinary bladder. Again the simple slide. You will find transitional epithelium, same picture. You can see the basement membrane columnar cell, the middle polygonal cells, and superficial the dome facet shaped cells. Below this, the lamina propria part, same. Then see the muscular layer. The muscular layer, you will find here the muscle layers in all directions, longitudinal, oblique, and circular. And all these are this type of muscles. Anyone? Which type of muscles? Yes, they are the smooth muscles, not the striated. Understand? Smooth muscles. So you will find all the layers of the smooth muscles. So very simple of the urinary bladder. Then another slide of the urethra. I am just giving you brief ideas because we will discuss again in the reproductive system. The male urethra and female urethra. In urethra, the proximal part of the urethra, I am using the word proximal part of the urethra, that is close to the urinary bladder. That part of the urethra is lined by transitional epithelium. I am telling you first the male urethra. The proximal part of male urethra is lined by transitional epithelium. The middle part and outer part, or the middle part of the male urethra, is lined by pseudostratified epithelium. And outermost part of the male urethra is lined by simple stratified squamous non-keratinized epithelium. So all the three types of epithelium. I will talk this again during the male reproductive system. So these are the different layers of the urethra. In female urethra, the epithelium are very simple, proximal part, transitional, and then the part of stratified squamous non-keratinized. So that is all about the excretory system. So we will show you the slide, kidney, ureter, and urinary bladder. I will show you these slides on the coming week. So revise this part and develop this habit. Need not to revise the theory part again in the practical class. So you can see the slide properly. And one thing which I had told you in the practical class also, but I am revising again. I will start giving you the one slide box in which all of you will take the slide, focus the slide. How you focus the slide, that I want to develop this habit. Because in university exam you will given the slide. That slide you have to own your focus. See the slide and identify the slide. No teacher will focus the slide. So you must develop how we focus the slides, how we keep the slide under the microscope. Always, whatever the slide, first of all, focus this properly light. Under first is the part of 4x, then 10x, and lastly 40x. And putting the slide, see the cover slip first. Second thing, don't go on the level because some slides are not properly leveled. Now you are in the stage that you can identify the slides. Because the leveling of the slide was done by the technicians, they may do the mistake. But you become doctor, you need not to do mistake. See the slide under the microscope. If you feel problem, ask us. So develop this habit also. Okay, very simple. And in the dissection, all have you seen the different vessels along the curvature of the stomach, branches of the celiac trunk? Everyone has seen? Okay. The pancreas not has been taught you, it will be taught you, but you can see along the pancreas also the structures. And there is a process, lower part of the pancreas, that is called uncinate process. Just deep to the uncinate process, there is the one artery. That is the part of superior colic mesenteric artery. And superior mesenteric artery you can see also very clearly. That is coming down, supplying the blood to the small intestine. And another is inferior mesenteric artery. Both are the branches of abdominal aorta. That is supplying to the part of large intestine. Now another thing, I had asked or told many tables, the splenic artery is tortuous. The answer is because it helps in the mobility of the spleen. The spleen movable, so it helps in the mobility of the spleen. Another is when you are surgeon, someone becomes surgeon, you do the surgery of GIT. With naked eye, how you differentiate that is, this is the large intestine, this is the small intestine. The differentiating point of the two, when you will dissect, you will notice this point. I am telling you first. In the large intestine, on its naked eye, there is the longitudinal bands, ribbon type bands. And these ribbon type bands are tinea coli. These are the collection of the smooth muscle fibers, and these smooth muscle fibers, they are collected on the anterior surface and inferior surface also. So the tinea coli are present on the large intestine. Besides, as you see the tinea coli, attached with the tinea coli, you will see the large number of the small folds. These are the appendices epiploicae. What the appendices epiploicae? The part of visceral peritoneum collected the fats, and these appendices epiploicae seen on the part of large intestine. So by seeing these structures, because histologically you can differentiate, but not in the operation theater, या काट के देखोगे. So you must be very clear about the gross picture also, how we can differentiate the large intestine from the small intestine. Okay, that's all.